Alright, so first off, the ligand or signal molecule from outside the cell moves and attaches to the G protein receptor molecule on the plasma membrane of the cell. Then the receptor sends a message across the membrane to activate the G protein. Then the G protein is activated by the phosphorylation of GDP into GTP. Then the G protein activates an enzyme called adenylyl cyclase. Adenylyl cyclase then releases a second messenger called CAMP by converting ATP. Then CAMP activates a protein cascade effect. Then the message will continue to move across the plasma membrane until it reaches the ER. After it reaches the endoplasmic reticulum, it then releases calcium into the cytoplasm. Alright, so first and foremost, the ligand, or signal protein, moves towards the tyrosine receptor on the surface of the cell. Then it attaches multiple tyrosines to each of its stems. These tyrosines on the stems of the protein activate other relay proteins in the plasma membrane, therefore initiating multiple signal transduction pathways simultaneously. The relay proteins are activated by the phosphorylation of ADP to ATP. Next, the signal moves into a cascade pathway much like the G protein event that we saw. Ultimately, the ER releases calcium into the plasma membrane, therefore ending the tyrosine kinase reception, transduction, and response. Breast cancer is caused by an irregularity in the signal transduction pathway of the cell reproduction system. So the result of this disease is an uncontrollable amount of reproducing breast cells, specifically for breast cancer. Now the ligand attaches to the receptor tyrosine kinase, as it normally would attach in any case. A massive amount of ligands keep attaching to the receptor to reproduce excessively. The over-amount of reproduction of breast cells ultimately leads to breast cancer. Now, to treat breast cancer, a drug called lapatinib treats this disease, and it is basically a drug that attaches to the receptor and stays attached to inhibit the breast cell ligand used in the reproduction of breast cells, therefore preventing the production of new breast cells.